presentation of polymer.
Oh,
Thank you.
that's very good.
Just like Catherine did. Bad news. Bad news is I will tutor you by the HIV. Okay, so my topic is pathogenic mechanisms of HIV disease. These are the authors. Here's the introduction. HIV was founded in 1983. It was isolated from lymph node. Do you know lymph nodes? It's an important organ for our immune system. We have some big lymph nodes here, yes, here and in the chest, or yes, and HIV will lead to the acquired immune deficiency syndrome, AIDS, which is AIDS, and it is a retrovirus. I will explain this later. I found it's difficult to introduce a reaction or mechanisms by words, so I think picture is more powerful. Here's the HIV variant, means HIV virus, and this red dot, and this red dot will crossing the barrier of the surface if you are cut or some wound. Here is a sample called DC. Here's a cell called DC. DC is a kind of cell with a lot of tentacles will help them to enter the human body. And after that, the HIV virus will conjugate with the T cells. T cells is important important cells for our immune system. They have some CD4 plus. It's a protein. They have a CD4 plus protein on their membrane, so the HIV virus will conjugate with the T cells. Actually, will connect with the cells, any cells with CD4 plus proteins. So that's why HIV will infect these cells. And after they connect with the T cells, they will inject their gene into the T cells. Their gene, means RNA or DNA, but for HIV virus, it's RNA. They will inject their RNA into the T cells. As you can, maybe not that clear, but here is the T cells gene. It's clean right now, but after being infected, their gene was there's a little part of the HIV gene on their own gene. So as they copy their gene, which is a necessary process for cells to separate, to split, they will copy the HIV's gene as well. So and then the HIV virus will be released by the infected T cells, and after that, notice here's the time, hours, days, weeks, and years. Our immune system won't be activated in days. So the virus. Here's the y-axis means the copies of HIV RNA. So you can see the amount of RNA goes up in the first maybe 30 days. Our immune system won't work during this time. They need time to generate the antibody of HIV. So after 30 days, maybe 20 days, they will, our body, our immune system generates the antibody successfully. The amount of the RNA of a HIV will decrease to this level, but not HIV was not destroyed. There are still some virus remain in our body. So this is HIV is affected. To illustrate to introduce the process, I need to introduce the central dogma of molecular biochemistry with enzymes. Central dogma of molecule. There's three paths. There are three paths. The general path is DNA can copy themselves, can replicate themselves, DNA to DNA. But there's another DNA to RNA and or RNA to protein. These are the general path, which many animals, or plants have the general path. But here's a special path. So the RNA can reverse this process is called transcription, and this process is called reverse transcription. So this is a special path, and RNA to RNA, DNA to protein, and the HIV's path is this, RNA to DNA. So the gene of the HIV is RNA, and they reverse transcribed into DNA. Okay, so there's another special. It says unknown, but we already know. I think the protein to DNA. I don't know if you know the BSE, bovine spongiform. Yeah, but mad cow disease. Do you know that?
Yeah.
Mad cow disease.
Oh.
Yeah. The pathogen is a kind of protein, and the protein can transcribe to DNA, and then from these parts. Yeah. So that's HIV's. That's the background knowledge of HIV. So, considering of the process of HIV infecting human body, there are 4 possible methods to cure HIV, to cure AIDS. First is furin inhibitors. Inhibitor means the one will stop you. Inhibitor means this inhibitor will stop furin and intrin. Furin and intrin happens here. The HIV virus will fuse the membrane of the cell, then inject their gene into the cell. So one possible way is to stop this process by furin inhibitors. The second one is integrase inhibitors. Integrase is a kind of enzyme, which helps the HIV virus to insert their genes into the T cells genes. Remember that after they inject their genes into the T cells, they will insert their own gene. They will insert the HIV's genes into the T cells genes. So this process needs the integrase inhibitors. So this is a possible way to stop that. The third one is reverse transcriptase inhibitor. Reverse transcriptase another enzyme to help the HIV with this process, the RNA to DNA. So there's another possible way to stop that process. And the last one is protease inhibitor. Even for HIV, they will go through this general path of the from DNA to RNA and to RNA to protein, and this protease inhibitor will stop this path from RNA to protein. So that these are the 4 possible ways to cure AIDS. That's all. Thanks.